Global warming. How can energy consumption be curtailed in order to address the biggest issue being faced by humankind in this century? One of the requirements is a highly efficient system unlike any other. In fact, there exists a highly efficient motor with energy conversion efficiency of virtually 100%. What is more, it rotates at a maximum speed of 1,500 revolutions per second, far faster than even the fastest Formula One race car engine. A bacterial flagellar motor with a diameter of only 40 nanometers, it is composed of protein molecules. 2 micrometer-sized bacteria rotate its flagella of 10 to 15 micrometers in length to achieve motility. The dynamic NanoMachine project, based at Osaka University, continues research to identify the atomic structure of the whole flagellum, which consists of filament, hook, and basal body. The aim is to create a foundation for bio-nanotechnology, friendly to both the environment and life forms. Bacterial flagella are composed of about 30 different proteins. First, a protein called Fly-F forms a rotor ring in the cell membrane. Fly-F self-assembles to form the ring, which then becomes the foundation for other proteins to attach, which in turn becomes the foundation for the attachment of others. The self-assembly takes place in methodical order through the accurate recognition of appropriate others. With a motor in place as the foundation, the filament that acts as the propeller is then formed of proteins called flagellin. With the help of a capping protein at the tip, flagellin molecules, sent out from the cell body through the central channel of the motor, stack up to form a helical tubular structure. The structure, revealed by electron microscopy, was very much like an artificial motor, with its stator, rotor, and bushing, and surprised the world. The I-Corp Dynamic NanoMachine project is one of the Japan Science and Technology Agency's international cooperative research projects. Joint research was carried out with Yale University's late Robert McNabb, who was professor of the Department of Molecular Biophysics and Biochemistry, and research faculty May McNabb, to identify the structure and function of various parts of the flagellum. Professor McNabb unexpectedly passed away. However, the research he had pioneered in flagellar protein export and his intent were continued by the Dynamic NanoMachine project, led by Professor Keiichi Namba. Self-assembly not only best characterizes the basis of life, but also represents indispensable technology for the future mass production of artificial nanomachines. "20 to 30,000 flagellin molecules self-assemble over 2 to 3 hours to construct a long filament. Electron cryomicroscopy, which allows high-resolution imaging of quick-frozen biomolecular structures at minimal radiation damage, was used to visualize the filament structure. Image analysis of electron micrographs of straight flagellar filaments led to a 3-dimensional image near atomic level. The central channel of the filament was extremely small, only 2 nanometers in diameter. Self-assembly of the flagellum, which grows out of the cell, always occurs at its distal growing end. The component proteins are produced inside of the cell and sent out to the tip through the central channel. The proteins are unfolded for insertion into the channel and then refolded at the distal end. Where then is the export apparatus? Research on flagellar protein export had been progressing extremely slowly until recently.
Basically, our bodies are made of proteins and nucleic acids that are expressed from genetic information encoded in DNA sequences. They form three-dimensional structures spontaneously. The one-dimensional blueprint starts taking a three-dimensional form at this point. Those molecules recognize partners and self-assemble to form large molecular assemblies, but it needs a device to bring them together to appropriate places.
There is something almost the same as flagella in shape, and with component proteins with extremely similar amino acid sequences. Called the needle, it is an organelle possessed by many pathogenic bacteria. It is used to secrete virulence proteins into human cells. Pathogenic bacteria such as E. coli 0157 produce many needle structures. It became clear that the export apparatus of the needle and the flagellum share a common mechanism. In other words, clarifying the mechanism of the export apparatus is likely to help us understand how pathogenic bacteria infect. The flagellar protein export apparatus consists of 6 membrane proteins: Flu A, Flu B, Fly O, Fly P, Fly Q, and Fly R, and 3 cytoplasmic proteins: Fly H, Fly I, and Fly J. The function of a protein can be revealed by observing cells depleted of the protein. Therefore, mutant variants missing those proteins were cultured, and it was found that virtually no flagella were produced. Bacteria could not swim if any one of the proteins was missing. It is thought that the 6 membrane proteins form the export gate. It has been thought that a complex consisting of 6 Fly I molecules is formed below the gate to export flagellar proteins together. It was also believed that the Fly I complex was attached firmly to Flu A and Flu B. However, the present study revealed that it was not the case. It was the finding by the McNabb group that many Fly HI complexes exist in the cytoplasm and also attached to the C ring. In other words, the Fly HI complexes assemble to the export gate for protein export and then detach themselves once export is completed. It was a dynamic structure. Fly I is an enzyme that hydrolyzes ATP. ATP is a chemical substance essential for cellular activities of living organisms. Energy is generated when it is hydrolyzed. Living organisms use it to maintain life. In the absence of Fly I, neither flagellar protein export nor flagellar formation takes place. It was therefore believed that protein export was driven by the energy generated by Fly I hydrolyzing ATP. Structural analysis of fly eye showed that it was surprisingly similar to the protein comprising an enzyme complex that synthesizes or hydrolyzes ATP. This ATP synthase uses proton flow across the cell membrane to synthesize ATP. However, the F1 portion, which was very much like fly eye, is also a motor that rotates by hydrolyzing ATP. As such, everyone believed that fly eye uses the energy of ATP hydrolysis to export flagellar proteins. However, Toru Minamino, leader of the nanodynamics group, made a discovery that turned around such a concept. He constructed mutant salmonella missing both fly H and fly I and observed its motility. Although slight, flagellar formation occurred after prolonged incubation. When cultured on soft agar, wild-type bacteria swim through the agar, spreading widely over time. Mutants that cannot export proteins cannot produce flagella, and so stay in one place.
This is missing fly H and fly I, but it's swimming a bit. This is missing just fly I.
The one without both fly H and fly I has spread a little.
The mutant missing both fly H and fly I spread slightly, a sign that the flagella were formed and the cells were swimming. However, the mutant missing just fly I did not spread at all. It was found that the motility of the mutant cells missing both fly H and fly I was greatly recovered when further mutations occurred in the proteins forming the export gate. This shows the amounts of proteins exported and secreted into the culture solution. The brightness of the band shows the amount. Mutations in flu-B and flu-A recovered the level of protein export by the mutant missing both fly H and fly I to a level even greater than that of wild type for some proteins. Fly H was somehow inhibiting protein export when fly I was missing. It showed that the export gate itself possesses the export function, and that ATP hydrolysis, which was thought to be indispensable for export, is not essential.
Up to now, it had been thought that the fly eye ATPase, the enzyme that hydrolyzes ATP, was extremely important for flagella formation. We found, however, that flagella can be formed even without fly eye. In other words, protein export takes place at certain probabilities. The process in which the ATPase is involved is important only in raising the probability of flagellar protein export, but not particularly important for anything else.
Besides ATP, another type of energy used to maintain life is proton motive force across the cell membrane. Flagellar motor rotation is driven by proton motive force as well. Group leader Minamino tried a reagent that shuts down proton motive force. When the amount of the reagent was increased, the level of protein export decreased. The energy driving the flagellar protein export process was also proton motive force. All of these experiments have shown that flagellar protein export occurs in the following manner. First, fly I associates with two fly H molecules. A flagellar protein to be exported also binds to them, and this ternary protein complex binds to the C ring and waits for the export gate to become disengaged. When the export gate is free and available, the ternary protein complex docks to the export gate together with free fly-HI complexes that are floating nearby or attaching to the C-ring. The gate is closed when it is free. However, it opens when the fly-HI hexamer ring complex binds to it and efficiently inserts the amino terminus of the flagellar protein into the gate. Later, fly-H and fly-I detach from the export gate through ATP hydrolysis. The export gate utilizes proton motive force to send flagellar proteins into the channel. It unfolds flagellar proteins into long stretched chains for export into the 2 nanometer channel. Once the whole chain is within the channel, it is transported to the distal end of the flagellum. Meanwhile, the fly-HI complexes that have detached from the export gate bind another flagellar protein and bring it to the waiting circle on the C-ring, repeating the cycle of binding and release for efficient export.
Being curious, finding it interesting, and wanting to know more is important. So is an open mind. Not to get caught up in conventional ideas, but to stay alert so that you see things that might be overlooked. Flexibility of the kind where you can always have an attitude of, hey, this is really interesting.
The flagellar proteins exported in this way bind one after another to the distal end. The flagellum becomes longer, and it eventually becomes possible for the bacteria to swim. The rotation of the motor is transmitted to the flagellar filament with a gentle helical structure to generate propulsion. The hook acts as a universal joint, so that the torque can be transmitted regardless of the orientation of the flagellar filament. Bacteria can swim about freely because of this. What is the mechanism here? The structure of the hook was studied in detail to learn the secret. The hook is a tube of 55 nanometers in length, in which roughly 130 hook protein subunits are bonded together. In order to clarify the complex structure of the hook comprised of many molecules, there was a need to combine X-ray crystallography with electron microscopy and image analysis.
It's very important to actually see molecular structures, because we are looking at a system of molecules. You can't really follow molecules with just logic and imagination. You can't see what is really going on unless you make the effort to see at the molecular level.
The characteristic of hook proteins to form tubular structures was very strong, and it was not easy to crystallize. The portions that caused the formation of the tube structure were identified. Hook protein fragments without these portions were expressed, and crystallization screening was repeated. Good crystals were finally obtained after about 3 years. The structure of the hook protein was finally solved after repeating diffraction data collection using extremely bright X-ray beams of Spring 8, the world's largest synchrotron radiation facility. At around the same time, electron cryomicroscopic analysis of a straight hook was carried out at Brandeis University. The data from Spring 8 and Brandeis were put together to build the atomic model of the hook. This is the atomic model of the hook. The D2 domains of the hook protein are strongly bonded to one another on the hook surface to form the right-handed 6-stranded helices, much like a spring, and they form a kind of mesh structure with the D1 domains on the inside. This is what brings about the rigidity against torsion. The large gaps between the six-stranded helices bring about the bending flexibility of the hook. A close look at the atomic model showed that there was a large difference in the length of the protofilament in the outer and inner part of the curved hook. The distance between the molecules is 3.5 nanometers on the inside and 5.9 nanometers on the outside, which is plus minus 25% extension and compression.
The overall structure is pretty much like a spring joint. And as you know, springs are very strong against torsion, but quite freely bendable. The molecules stack up in such a way to form something exactly like a spring joint.
Furthermore, dynamic slipping between molecules in response to the extension and compression of the protofilament was revealed at the interface between D1 and D2. It was this dynamic structure that allows motor rotation to be transmitted smoothly to the helical propeller, regardless of its orientation. The length is also important for the hook to be able to work as a universal joint. While there is a large variation in the length of the flagellar filament, the length of the hook is almost constant at 55 nanometers. Mutants with hooks longer or shorter than 55 nanometers cannot swim properly. So how then do hooks have the predetermined length of 55 nanometers? It was known through recent studies that Flu-B, which composes the export gate, and Fly-K are involved in hook length control. It was gradually revealed that Fly-K is also exported and works as a molecular ruler that measures hook length. The central channel of the flagellum is only 2 nanometers, barely large enough for one unfolded protein chain to make it through. If Fly-K remained in the channel, hook protein would not be able to go through, and the hook would not be able to grow. When the hook has not yet reached the right length, Fly-K passes straight through and is excreted. The hook assembly continues until the next Fly-K molecule is exported. When the hook reached roughly 55 nanometers, interactions between the C-terminal domains of Fly-K and Flu-B take place, triggering a structural change of the export gate. This switches the export selectivity from those making the rod and hook to those making the flagellar filament. However, there was a phenomenon that could not be explained simply by this mechanism. While the mutant missing Fly-K makes very long hooks, many of them are actually 55 nanometers in length. How are hook lengths determined, even when the ruler is not working? The dynamic measurement group measured the length of the hooks made of mutant hook proteins and noticed that hook formation was slow and that many of the hooks were shorter than normal. It takes time for the hooks to grow with these mutant hook proteins because they do not easily bind to the hook. The fact that short hooks are produced due to slow growth is an indication that something must be measuring the time of hook assembly. It was thought that hook protein export slows down to virtually stop hook assembly after a certain period of time passes. And it was likely that this was the reason why hook length can be precisely controlled, even with a few times length measurements by Fly-K. A cleavage within the cytoplasmic domain of Flu-B is necessary to stop hook growth and begin exporting proteins for filament formation. Group leader Minamino had found that the speed of hook protein export slows down by such cleavage. It was amid such circumstances that Yale's McNabb group found that the cleavage of Flu B occurs by itself, even in the test tube. These findings combined show that there is a mechanism that controls hook length to an extent, even without Fly-K's ruler function. There appears to be a timer of sorts that controls the time period of rapid hook assembly.
In the model that we proposed, several Fly-K ruler molecules are secreted out of the cell, giving them the opportunity to measure hook length while a flagellum is constructed. Slowing down the speed of hook assembly would enable the Fly-K ruler to make precise measurements. We think that autocatalytic cleavage of Flu B is working here as a molecular timer.
The mechanism that determines hook length is as follows. First, hook protein molecules are efficiently exported, and the hook becomes nearly 55 nanometers in length. The autocatalytic cleavage of Flu B slows down the export of hook protein. Then several Fly-K molecules are exported to measure hook length. In other words, the cleavage of Flu B works as a molecular timer that controls the speed of hook protein export. As you can see, the precision mechanisms for constructing biological nanostructures are becoming clarified. The Earth simulator is a supercomputer that Japan is proud of. Professor Namba says that even the Earth simulator is a far cry from the functions of living organisms that evolved over some 4 billion years.
The brain of a small fruit fly uses energy in the microwatts for complex flight control and visual information processing to find and fly to food. I don't think a supercomputer could yet simulate what the fruit fly brain does, even while using megawatts of energy. The difference of more than 10 orders of magnitude in the level of energy used is an indication of just how incredible biological systems are. It even exists in bacteria. The flagellar motor and protein export apparatus use proton motive force, or mechanisms that utilize the flow of protons at extremely small energy, close to the thermal noise level. Understanding the basic physical mechanism behind them will bring about the time when it can be actually utilized for engineering. It is work to achieve the dream of resolving global environmental and energy issues. That is how big it is.